Okay, so let's start with the central connector, shall we? The central connector, which is the sugar, which is going to be the ribose sugar. Okay? Now, here we have the, you know what? Let's make them two different colors. Here's the DNA, and here's the RNA. Why does the RNA get to be blue? Yeah, it doesn't deserve to be. Here. And the RNA. Right? Now, I'm going to draw both pentose sugars. I'm going to tell you why we call them pentose. I'm going to ask you, what difference do you see in them? At this point, you have a good eye. There's no oxygen group on the sugar or the ribose sugar that makes the DNA, while on the RNA, there's going to be a hydroxyl group. Remember, hydroxyl, OH. So it has an oxygen, the RNA is sugar. The RNA ribose has an oxygen, the DNA lacks it. Okay? Now, before we talk about why, let us talk about the numbering system of the ribose sugar and why it's important, right? So the ribose sugar characteristics, number one, the numbering system. When we count the carbons, we're going to say 1 prime, here's a carbon, 2 prime, 3 prime, 4 prime, and 5 prime. The same thing goes, of course, for the RNA, right? So why are they, first of all, why is it called prime? Why? Prime, right? Quite basically, the nitrogen base, remember, the sugar is a central connector. The nitrogenous bases are going to connect to the number 1 prime carbon, right? Now, the nitrogen bases are rings themselves, just like this. And so we count them 1, 2, 3, 4, 5, 6, for instance. So to avoid confusion, we simply refer to the numbers on the sugar, on the ribose sugar, as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And great. So we spent a lot of time talking about numbering a goddamn sugar. Now what? Now all of them are important except for 4 prime. We don't care about it. How are they important? Now remember, we said that the sugar is a central connector. 1 prime is going to connect to the nitrogen base. 5 prime is going to connect to the phosphate group. 2 prime, well, basically 2 prime tells us the difference between DNA and RNA, right? So 2 prime, if there's an oxygen group, then it's RNA. If it lacks, if it's deoxygenated, then it's going to be a DNA. What about 3 prime? 3 prime, basically, this structure right here, remember we said this is going to be a nucleotide. If there's another nucleotide and it wants to attach this nucleotide to make an oligonucleotide and then a polynucleotide, then a DNA, they're going to attach themselves on the 3 prime, okay? And we're going to talk about that, don't worry. So this is the numbering system. Number two, okay, so central connector, right? Central connector, right, great, okay. Number 3 is, why does the DNA lack an oxygen on the 2 prime end? Quite basically, listen, this is what happens. Here's an RNA, right? Here's an RNA. And the RNA, this is the backbone of the RNA. So basically, the backbone is going to be the sugar and the phosphate, right? Now, because it has an oxygen here at the 2 prime end, here's the 1 prime, by the way, and 2 prime, 3 prime, 4 prime, and 5 prime. Now, because it lacks an oxygen group at the 2 prime end, what's going to happen is that it causes self-cleavage. It is very reactive. The mere presence of this oxygen right here is going to cause the RNA to go and attack itself and undergo something called hydrolysis. Now, hydrolysis, remember, so water breaking. This is an RNA, and I put a drop of water on it, it breaks, right? And so you'd imagine that the RNA is not going to survive for a very long time. Now, what about the DNA? Well, see, the DNA has to survive for a very long time. As long as the cell is living, the DNA must be very chemically stable. How do we achieve that? Well, we simply remove the oxygen that was present here. And that's why that's why DNA lacks oxygen. Okay? See, DNA is the genetic blueprint of the cell. It has to survive the entire life of the cell. And sometimes, if we're talking about germ cells like spermatocytes or oocytes, it has to even outlive them. It outlives the original human, right? And that's why it lacks the O2 to keep it chemically stable. What about RNA? We know that the cell has to be energy efficient, right? The cell is a very stingy subject, by the way. It does not like to use energy when it can, conserve it. So why would it make something? Why would it make a structure for a couple of minutes, half an hour, whatever, and then that structure goes and degrades itself? Well, think about it this way. Suppose this is a cell, right? And here is a nucleus, and of course, the cell is very happy. Why is it happy? It has its RNA, it has a messenger RNA, and this mRNA is making proteins, right? Now, suppose that this mRNA doesn't degrade itself. Suppose that it lacks an oxygen group like the DNA does. So the mRNA, the only thing it knows to do, or one of the most important functions of it, is to make proteins. And it's going to keep on making proteins and making proteins and making proteins, and eventually the cell dies. So RNA is chemically unstable. It has self-cleavage. It destroys the self. DNA, on the other hand, has to live for a very long time. Okay? So why did we spend so much time understanding the deoxyribose sugar or the ribose sugar? Oh yeah, another point before I go there. Listen, the ribose sugar is also called a pentose sugar by some textbooks. Penta, by the way, pentose or penta means 5. Penta means 5. Why is it 5? Because it has 5 carbons, right? It has 5 carbons, so we call it pentose. Of course, pentose simply refers to how many carbons it has. There's a lot of sugars that are also pentose. Xylose is considered pentose. Arabinose is considered pentose, as is ribose, right? And notice the ose suffix. Xylose, arabinose, ribose, right? So it's a suffix, means they're sugars. That's all. Okay, so again, why did we spend so much time talking about the ribose sugar? Imagine this. There are some tumors, or let's take an example about the herpes simplex virus, right? So herpes simplex virus, it's a DNA virus, right? And it's the way, by the way, most species, or all of them in fact, except if it's an RNA virus, make their DNA is extremely close to how we make our DNA, right? So, and this herpes simplex virus, it infected this person. And now we go and we give something like vidarabine. Vidarabine is a drug. The difference, it resembles ribose sugar, but it actually contains arabinose sugar. The arabinose sugar doesn't look quite like this, it has maybe the structure is different, maybe it lacks an oxygen group here. And this is just as an example. We're going to talk about this when we reach pharmacology, right? But just bear for me for a minute, just bear with me. So suppose that we give a drug and we remove the hydroxyl group from here, we remove the oxygen and we left this. And so the virus would come and it's trying to build its own DNA and takes it up, and by the time it takes it up and it incorporates it into its own DNA, and now it's going to take another nucleotide, right? And it wants to stick it on here. But wait, where's the oxygen? This is called chain termination. This is one of the ways where we kill viruses, and by the way, one of the ways where we can use drugs in treating certain leukemias. Stuff like cytarabine, right? Cytarabine is a drug that's used in acute myeloid leukemia by interfering with the DNA synthesis and replication. Okay, basically, again, cytarabine, vidarabine, fludarabine, right? Now, listen, look at the suffix, arabine, 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 right? So basically, the sugar is not a ribose sugar in these drugs. They just resemble the ribose sugar, in which case, if it's a cell that has cancer, for instance, like an acute myeloid leukemia or chronic lymphocytic leukemia, they're going to take up that sugar into the DNA, because remember, cancer cells, all they want to do is divide, 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 right? Now, if you cause chain termination, if you prevent the DNA from, elongating itself, then the cell never becomes, and you try to control the cancer. Okay? I think that's all we have to talk about for the sugar. Okay? Yeah. Okay. Now it's time for the nitrogenous bases. The NB, right? Again, nitrogenous bases. Why do we call them nitrogenous bases? Well, nitrogenous because they contain nitrogen at different carbon numbers, and bases because, remember, they accept a proton, and we call them bases. Okay? So, what do we need to know about them? Nitrogenous bases. They're subdivided into purines and pyrimidines. There's a pyramid. Okay, I'll write it down. I don't want to be lazy. Here's a pyrimidine. Right? Here's the pyramid. Here's a pyramid. Okay, anyway. So, purines and pyrimidines, what's the difference? A purine has 2 rings. A pyrimidine only has 1. So, how are you going to remember this? Easy. Pu, here's 1 ring. Rene, and here's another ring. So they have 2 rings, the purines. What are they? They're adenine, or it could be guanine. What about the pyrimidines? The pyrimidines have only 1 ring, and the way I remember this is, if that has 2 rings, this has 1. Okay, it's a pyramid, 1 ring. So anyway, cytosine, it could be uracil, and it could be thymine. By the way, don't say thymine, because that's a vitamin, okay? Thymine, thiamine. They're very close to one another, but anyway, you get the idea. Now, how do students, a lot of them remember this? Purines, they say it's pure as gold, and the pyrimidines, they simply say cut. Okay? Now, unfortunately, at this point, me and you are parting. It's time for the tablet. It's going to make things faster. It's going to make explaining this much more easier. Okay? So to the tablet. Easier. Okay. So a quick recap on the nitrogenous bases. In DNA or in RNA, there are going to be purines, right? And there are going to be pyrimidines. How do you tell the difference? First of all, before we go there, the difference between DNA and RNA, there's only 1. In DNA, it's going to be thymine. In the RNA, it's going to be replaced by uracil. Okay, so DNA contains thymine. RNA, instead of thymine, it has uracil. Okay, and that's the only difference between the nitrogenous bases. Now, the nitrogenous bases themselves, they could be either purines, which means there are 2 rings, right? Or they could be pyrimidines, meaning that there's only going to be 1 ring. Okay, and here's a table to quickly recap it. So adenine is going to be a purine. Guanine is also going to be a purine. Cytosine, thymine, uracil, and all of these 3 are going to be pyrimidines. Notice that thymine is only present in DNA, while uracil is only present in RNA. And here is a visual so you can understand it really. So purines, again, they have 2 rings, 1 ring, 2 ring, and pyrimidines, they only have 1 ring. Now, one of the things you're going to encounter in the exam is, well, again, the purines. By the way, there is students use it. They say that purines are pure as gold, to remember the purines, which are adenine and guanine, right? So pure as gold, adenine, guanine. Pyrimidines, again, only one ring. We've got cytosine, thymine, and uracil. And the mnemonic is cut, right? So cytosine, uracil, and thymine. Now, why is this important for us to know? Now, one of the ways the USMLE asks about it is that they could present you with a nitrogen base, and then they expect you to know which nitrogen base it is, okay? So again, if it's two rings, it's going to be a purine. If it's one ring, it's going to be a pyrimidine, and that's nice, right? So how do you tell? It's very easy to tell a purine from a pyrimidine, but how do you tell two purines from one another? For instance, over here we have the adenine and we have the guanine, and they're going to give you the whole chemical structure of it if they ask about it. First of all, I want you to forget about all of these. They are not important, okay? Not important. What is important is the carbon number 6 position, which is, in the case of adenine, this one right here. So in purines, if you see an amino group, this is an amino group, by the way. There's an amino group. If you see it at the 6 carbon or the 12 o'clock position, some examinees call it. So here's carbon number 1, 2, 3, 4, 5. This would be carbon number 6. And you look atop it, and if you see an amino group, you know this is going to be adenine. What about guanine? So great, here's ring number 1, ring number 2. Where's carbon number 6 or the 12 o'clock position? And here it is. What does it have? Does it have an amino group? No. And therefore, this is going to be a guanine. Okay. What about the pyrimidines? So what about the pyrimidines? Pyrimidines, again, they have one ring. So here's one ring, one ring, one ring. How do we tell them apart? How do you tell cytosine from thymine from uracil? First of all, again, we look at the 12 o'clock position or carbon number 6, right? If it has one ring and has an amino group on it, here it is, here's the amino group, then this is going to be cytosine. We do not care about all these other structures. Now, what if it's, okay, so basically uracil and thymine, they do not have an amino group. So how do you tell thymine and uracil apart? Simple. Thymine has a methyl group, and usually it's written like this, HC3, right? So if you see a pyrimidine, meaning there is one ring, and you see an amino group, then this is cytosine. If you see a pyrimidine and you see, if you see one ring, it means a pyrimidine. If you do not see an amino group, then that means that could be either uracil or thymine. How do you differentiate between these two? Well, again, you look at the carbon number 5. Here, you're going to see a methyl group, and that's all. So the only difference between uracil and thymine really is the presence or absence of the methyl group. So now we're faced with two questions. Number 1, why is there no thymine in RNA? And number 2, why is there no uracil in DNA? Let us start with the simpler explanation, which is why is there no thymine in RNA, right? Now, chemically, uracil is like thymine, but without the methyl group, okay? And the reason that RNA really does not contain thymine, first of all, thymine making uracil is simpler. It's cheaper. Remember that we said that the RNA, because it has it has an oxygen at the 2' carbon of its sugar, of the ribose sugar, this means that's going to be degraded quickly, right? And therefore, the cell, being energy efficient, doesn't really want to use extra energy putting methyl groups on every base, okay? And the second reason is for speed. Because RNA is going to be produced, it's degraded constantly, so of course simplicity is going to be always favored. Okay, now that answers the first question. Okay, it's very easy. That's why we don't have thymine in RNA. Basically, energy, efficiency, and speed. So let's go to the second question, which is why is there no uracil in DNA? So there are 3 reasons for this. Number one is something called cytosine deamination problem. Let's leave it for now. Number two is stability. Why? Because thymine, remember, we said thymine has a methyl group, right? And what this methyl group does is it makes DNA more stable, it makes it more resistant to mutation, okay? Again, we said that DNA, it's the genetic blueprint of the cell, and therefore we have to protect it at all cost. And which explains really point number three, which is longevity versus disposability. DNA is a permanent storage, and therefore it needs extra protection. RNA, remember, it's a temporary copy, it's a temporary message, and therefore we use cheaper material to make it, okay? Like Uracil, which in fact is more stable. Now, the important point here, the one I want to talk about, is the cytosine deamination problem. Now, remember people, we said that to always look at the title, it explains a lot. Deamination. Remember when we said deoxy and we said that the DE simply is a prefix that means lacking or without, right? And amination or refers to the amino group, which simply means NH3+. Okay, so deamination means we take the cytosine and remove the amino group, and now we are left with something called uracil. And that is quite problematic. So why is this problematic? Listen, let's draw a strip of DNA, right? And it has nitrogen bases. So it has adenine, guanine, cytosine, thymine, adenine again, for instance, cytosine, thymine, okay? Now, sometimes a mutation occurs that is called spontaneous cytosine deamination, right? And we talked about this. We said the cytosine, it spontaneously loses the amino group and it becomes like this, right? And suppose that the C here underwent that, and now you have a uracil. And so we have these little enzymes, clipper enzymes, that they come in and they see that, you know, there's a uracil here, and uracil shouldn't be present in DNA. They cut out this portion, they remove the uracil, and then they reinsert the cytosine again, and everything's fine. Okay? Now, suppose that uracil was in fact present in DNA. So let's draw another strip of DNA, and let's put nitrogen bases, adenine, for instance, guanine, thymine, cytosine, uracil, guanine again, cytosine, uracil, for instance, okay? And so let's suppose that this C over here underwent spontaneous cytosine deamination, and now it's transformed into a U. And so this repair enzyme, it comes in, right? And, you know, it's checking, okay, you're good. It sees a uracil, fine, you're where you're supposed to be, guanine, and then it comes here and sees a uracil, okay? And since, well, it thinks to itself, well, uracil is normal, it's present, and so it just keeps on moving on. And it does not fix this uracil right here. So what happens is, now this uracil here is going to be a hot spot for mutation. It can cause severe problems, okay? And this is what is called cytosine or spontaneous cytosine deamination. And that's why one of the more important reasons why we do not use uracil in DNA. So, can uracil be present in the DNA? In fact, it can. Two problems, or two ways it can occur. First of all, is the spontaneous cytosine deamination. Again, we said cytosine loses its amino group and then becomes uracil, and we talked about this. Number two is the replication errors, right? Basically, one of the enzymes that's involved in DNA replication is called the DNA polymerases. It's a very important enzyme, and sometimes it makes a mistake and it incorporates deoxyuridine monophosphate instead of deoxythymidine monophosphate. Okay, so this is why, yeah, uracil can sometimes be present in DNA, and it is also very problematic. It causes mutations if it's not fixed. However, it usually is because remember, cells always have specialized repair systems. Okay, in this case, it's something called a UDG scan or a uracil DNA glycosylase. Not important for us here. We're going to talk about this when we go over mutations. All right, but basically, it removes the uracil base, and then base excision repair enzymes come in and they fix the problem. Okay, so far we went over the ribose sugar and the nitrogenous bases. Now, where do we go from here? Well, we want to connect them. Remember, we're making a DNA, so we want to build a nucleoside. To build a nucleotide, then to build an oligonucleotide, then a polynucleotide, then a single-stranded DNA, and eventually a double-stranded DNA. So, back to the tablet. Okay, so now we have our sugar, our ribose sugar, and we've got our purine and our, of course, pyrimidine, right? And now we want to connect these two with one another to form something called a nucleoside. Now, the way I'm going to do this is that I am going to draw for you a certain structure. And before I say it, I want you to pause and think about the structure I have just written, okay? So, basically, first of all, what happens is, on this sugar right here, remember 1', 2', 3', 4', and 5'. Where does the nitrogenous base, whether it could be a purine or pyrimidine, where does it link? It links at the prime number 1, and the linkage, if you want to know it, is called beta-N glycosidic bond. Okay? Now, to the juicy stuff. What's this? Well, it is the 12 o'clock position, or the carbon number 6. Remember? 1, 2, 3, 4, 5, 6. Is there an amino group? No. How many rings? It has 1 ring, it has 2 rings. So what is it? And don't say guanine. I mean, the nitrogen base is guanine. However, remember, it's going to be connected at the 1' to the ribose sugar. And so, what's it called? Well, you can't really tell yet. Why? Because it could be either guanosine or it could be deoxyguanosine. So, how do we know? Well, if there's a hydroxyl group at 2', then you know that this is an RNA, and so it's called guanosine. If there is no oxygen here, only a hydrogen, at the 2' on the ribose, it's called deoxyguanosine. Okay, what's this? In fact, you know what? I'm not going to draw anything. What is this here? This is 2'. Here, again, 1', 2', 3', 4', 5', just so you don't forget. And this is the carbon number 6 on the nitrogen base. What is it? And listen, I know there's going to be chemical structures here. However, remember, there's nothing on 6, there's nothing on 5, or if simply does something like this, this is a uracil. Okay? The nitrogen base is called uracil. The whole structure here, since it's an RNA, is going to be called uridine. Okay, let's draw another one. Okay. Here. Here's an amino group. What's this structure? Well, since at the 12 o'clock position, or carbon number 6, first of all, 2 purines, 1 purine, 2 purines. There's no oxygen here, so it comes from DNA, so it's deoxy. Deoxy what? What's the structure? And it's adenine. So it becomes deoxyadenosine. Right? Okay. What about this? What's this structure? Here. I'll tell you what the structure is. This structure is wrong. Now, you may be tempted to say something like deoxythymidine or thymidine, but remember, this comes from RNA, and thymine is never present. And so, this is a wrong structure. Okay? It's never present. The thymine is never present in RNA. This is an RNA, right? Now, if I did something like this, if I removed the methyl group here, and I gave something like this, now this is going to be uridine, right? Okay, what's here? One ring, two rings, and there's an amino group. So this is adenine, right? Now, since it's connected and the number 2' has its oxygen, has a hydroxyl group, and therefore this is going to be called adenosine. Okay, what about this? What about this? Well, you know, what's this? This is deoxycitidine, right? Now, I'm going to draw them again. I'm sorry, I know, but it's very important that you understand this. Now, let's start with the DNA, then we're going to turn it into RNA, right? Now, what's this? This is deoxy, remember, because there's no oxygen on the 2', so it's going to be called deoxyadenosine, right? Okay, what about this? What's this? So, on the 2', there's no oxygen, so it's deoxy, 1 ring, 2 ring, no NH2 group, no amino group, so this is going to be deoxyguanosine. Great. What about this? What's this going to be? It's going to be, yeah, deoxycitidine. What about this? You're correct. That's going to be deoxythymidine, or sometimes we simply refer to it as thymidine, right? Okay. What about this? And I hope you didn't say deoxyuridine, because remember, uracil, this is uracil, because remember, there's no NH2 group, and there's no methyl group, so it's going to be uracil. However, technically, it could be found in DNA, but it would be an error and it would be wrong, okay? So the correct manner to write this would be to add an oxygen group on the 2', right? So it becomes uridine. Great. So what if I do this? What's this now? Remember, now it's part of RNA because there's an oxygen here. And it's going to be called adenosine, right? So adenosine. What about this? Now it's going to be called guanosine, right? Now, notice in the purines, there's the sine suffix. What about this? 1 ring, amino group. This is going to be called cytidine, right? What about this? Careful now. This is thymine. It cannot be present in RNA, and so the structure is wrong, right? So, and this, of course, is going to be uridine.